All right, today I'm going to start off with Punnett squares. We already talked in the screencast about Punnett squares earlier. Going to give you a little introduction to mono hybrid crosses. Remember, mono is one. Hybrid is when you get something that has two different letters, right? You've got a heterozygous. Make sure you know that term heterozygous. Make sure you know the term homozygous. If it's heterozygous, it's just heterozygous. It's not heterozygous dominant or heterozygous recessive. The fact that it's heterozygous says it all. There's two different letters. You can have homozygous dominant, and in that case, you'd have two dominant alleles. And you can have homozygous recessive, and in that case, you'd have two recessive alleles. But if you're talking about heterozygous, it's just heterozygous. We left off, I think, talking about test cross. We actually got to this idea that Mendel talked about the law of independent assortment. Now that we've done meiosis, we can talk about this a little bit more better. Yeah, that's good English. I think it'll be a little more easier to understand. So, I actually don't want that picture. I want, let's see, this one, maybe? Yeah. Okay. So, monohybrid crosses, obviously one trait, but when you start looking at more than one trait together, and you look at the probability that you get two particular traits together, you can do what's called a dihybrid cross. So two traits that you're crossing. And when you do that, you come back with different ratios. The Punnett squares will give you different ratios, or I shouldn't say different ratios. They'll give you ratios, and then you can compare those ratios to what you actually observed. And so Mendel, in his experiments, he always used, and he didn't know this at the time, but he always used traits that show independent assortment. And we're going to go through a dihybrid so that I can show you that idea. Nice phrase. The idea is that do alleles for one character. So if I got one trait, does that trait, does the chromosome for that trait go into a cell, a sperm cell or an egg cell, dependently or independently of alleles for another? In other words, I have two traits. Maybe I have round seeds and I have yellow. What are some of the? Here, let's see what they use, because that's what we're going to use. Oh yeah, round seeds and versus wrinkle, and yellow versus green. Those are two different traits that pea plants exhibit. Okay. Do you see those traits going together kind of as a package, as a unit? Or do you see those, it doesn't really matter if you get yellow pod or yellow pea color. It doesn't have anything to do with whether or not you get round or wrinkle. Okay, are those two traits going together as a package, or are they going separately, independently of each other? And so what I'm going to do is, let's see, what am I going to do? I'll just show you this. Let's make this a little bigger, though. This is really small. Well, that didn't help. It's supposed to work that way, right? So nerd. Oh, that didn't work either. What the heck happened? Who are you calling nerd?
Because I put on glasses and I'm a nerd.
Oh, come on. Don't hate on the glasses. Come on.
I wear glasses as well.
He feels like a nerd.
All right.
Like a nerd, bro.
This is not working well.
I think it means we're done with this.
Oh, Evelyn. Oh, Evelyn. There it is. Yes. Now it should get bigger, right? I don't think so.
When you were hitting the mic, it was zooming out. Whoa.
Yeah, but it's not supposed to. I know We've done this before. It's taking,
Oh, it is.
yeah, it's zooming in.
Why? Very.
Oh, that's as far as I can go. It's like, that's it. All right. Oh my God, now I'm not going to ever be able to actually see anything else. Do I have a... No, I don't. Oh, how am I going to hit that?
Intermission, guys.
There, I got it. Ha! Okay. So, I'm starting with two characteristics. I'm starting with yellow versus green, and I'm starting with round versus wrinkle. Okay? Pea color. Pea pod, or peas. And I'm going to start off with true breeding. And so true breeding means I always produce the same outcome. So I have yellow and round seeds, and I also have green and wrinkled seeds. And then I cross those two together. So I do that cross fertilization, and I get an F1 generation. So I know, because these are true breeding, that the offspring, the F1 generation, are going to be heterozygous for both traits. Okay, we're using big Y for yellow here because yellow is a dominant trait, and we're using big R for round because round is a dominant trait. Okay, so they're all heterozygous for both traits, for yellow and for round. And then I let these guys, I can either let them self-fertilize or I can cross-pollinate. Either way, I want to see what kind of seeds I get in what percentages I see in my offspring. So there's two predictions. One is that it's called dependent assortment, meaning that if yellow goes into one sperm cell, then also going with it is going to be that round coat, or that round seed texture. Okay, that's dependent assortment. Independent says no, it doesn't matter. If you're yellow, you could be wrinkled or you could be round, but they're completely independent of each other. So you get different numbers, in other words. And when Mendel did this, he actually found results that are below here. And that shows you that when you do the Punnett square, you see that you have results that support the independent assortment hypothesis. Now, I think it would be just better. Hold on a second. I got to go back out to have you do this problem. So we'll do that as soon as it figures out what we're doing. Okay, cool. So we're going to do a dihybrid cross. I'm going to have you do it either on your note packet, or probably you want to do it on a piece of paper, another piece of paper.
That's an astute observation.
Yeah.
Okay. We have two parents, and our parents are both big Y, small y, and big R, small r. We're going to do a dihybrid for this. Now, when you do a dihybrid, you have to figure out, when we did monohybrids, it was pretty easy because you're only dealing with one trait, right? So you were dealing with, Y, big Y, and small y. Okay? And so when you were doing that, you would just put one letter above each box, like that. You're going to do the same thing in this particular example, but now you're doing it for two traits rather than one. So in a dihybrid where you're dealing with two parents who are heterozygous for both of those traits, what's going to end up happening is you're going to have not 4 squares. I heard it. You're going to have 16. Oh my gosh. So I need to draw a 4 by 4. 1, 2, 3, 4. Okay. This is why I suck at art. I can't draw it. Oh my god, that's terrible. Anyway, yeah. All right, whatever. So now for each parent, I'm gonna have one parent here, and I'm gonna have one parent up here. I need to figure out what the allele combinations are that can be passed on in the sperm or the egg cells. Okay. So for this first parent, what could I pass down in terms of yellow versus green color? What letters can I pass down? I could pass down big Y, or I could pass down little y. Okay. But we need to also look at the second trait, which is round versus wrinkle. What could I pass down? Round, R, big R, or I could pass down little r, which is wrinkle. Okay. I need to know the combinations of how many of those I can have. So I could pass down, what do we say? I could pass down a big Y, and that would be the only allele for that trait, right? Because we're only going to pass, remember, those homologous chromosomes separate. And we only give one of our homologous chromosomes. So one of our homologous chromosomes here is big Y, and one of them is small y. And I just chose big Y. What about for my second letter? What could my second letter be? Little or big? Has to be the different letter. R. Big R.
Wait, why would you?
Yeah, clearly.
We have two traits, and we're picking letters. We're picking one letter for each of them. So you should have 2 letters per line or per column. So I just did big Y, small y. Let me move this around here. All right, give me another one.
Small y, big R.
Small y, big
I
R.
was literally just thinking that too. That ass.
Small y,
It's not many, honestly.
That ass.
small y, big R. That is another one. Next.
Small.
Big
Y,
R.
Small R.
All right, if we're doing big R, that big R, we're going to keep big R second because just we started with Y, so we have to use Y first. So what's Y? What kind of Y? Big Y. Okay, let's use a big Y. And then what? Small R. Yeah, that would work. What's my last one? Small y and small r. Okay.
How did I end up with
When you have 2 letters like that, for 2 traits, you're going to have a total of 4 possibilities. Now, that's one way of doing it. The other way, for you mathy people, is you can use, get exactly the same thing. If you're not a mathy person, you can ignore this. Anybody remember FOIL?
Yeah.
Oh, yup.
Goated.
Okay. All right. So FOIL refers to first, outside, inner, and last. So you put parentheses around your Y's and around your R's, like I did here. And I look at the first letter of each parentheses. What's the first letter of the first parentheses?
Big Y, big R.
Big Y. And the first letter in the second parentheses is big R. Then I do outside. First letter on the first parentheses, big Y. And I look at the outside, the very outside on the other edge, it's a little r, outside. Then I do inside, small r, big R. And then I do the last ones. Okay. By doing it this way, you're assured to get all the combinations. But you're always going to have a max of 4. Okay. Let's go. Okay, so now the next thing we have to do is combine them. So to combine, we're going to stick with the convention of Y's first, and obviously dominant letter first. That's my problem too. I don't think I made it big enough. Okay. If I let's see if I do this one down. I'm gonna go down one row. Again, always put the dominant letter first, even if as you're pulling it across, it would normally go before the big letter. You always put the big letter first. It's just a standard that we use. Okay. Okay. Can you fill it out? See what you get? All right, fill it out. This will be a question on your genetics problems, which replace the essay for this unit.
Wait, when's the test?
February 9th, I think.
What is the score?
I don't know. I know conferences are the 29th of February.
I don't know if she knows that. But she always did the big search.
When are we supposed to go?
I don't know.
Oh, I messed up.
You know, next year school starts on the 12th of August.
Actually, is the big letter always first?
What's that?
Big letter's always first, right?
Big letters always go first, yeah.
Big
And if you started with Y's, always leave it Y's first and then R's.
Wait, how do you know to start doing for the next year, though?
You can look it up. It's already published. It's out on the web.
Like the 14th. 16th.
Let's solve this.
Is that tough?
That is.
Yeah.
Like, crazy. We're literally not gonna. We're done.
Started the 15th last year.
I'm just kidding. That was so tough on it.
Exactly. I know.
Day before Memorial Day.
It's okay.
All right, once you have this done,
How does it feel like Trump's a big man? Big man on campus.
can I erase this? Is this okay to erase this? It'll be in the recording if you really want to see it.
Almost a big man.
You're good?
on campus.
Okay, so I'm going to clear the screen. Now I want to know. I want you to break it down for me. I want to know how many are yellow. Oops, that's really not very well. How many are yellow and round? How many are yellow and wrinkled? I'm just going to say rink. How many are, nope, that's not how you spell green. Green and round. And how many are green and wrinkled? Right? Figure that out. I'll give you a hint. They should add up to 16. Yeah.
So for it to be green, it has to have 2 small yellows.
Has to have 2 small Y's.
And
Yep.
for it to be wrinkled, it has to be 2 small R's.
2 small R's. Right. That is correct.
There's 4. Those are the 4. Okay.
Yeah, but what does what?
So as long as it doesn't have like a dominant gene, then it will. Okay, so like you see how it's like big Y, big R. If it has 1 Y or 1 R, then it's gonna be
Then
yellow.
it's gonna overpower.
Yeah.
Okay.
It has to have.
Has to have one
I was
dominant.
asking, I didn't know if the
Yeah, yeah.
big R was round or the
Oh
little.
yeah. Okay.
Sorry, yes, it does. The big R is round.
And then
Yeah.
the big. Okay.
Let me just say that. Oop, that's not what I wanted though. I wanted the other one. So big Y equals yellow, and big R equals round. Have to have two recessive alleles to show the trait. Otherwise, it's going to show the dominant allele if it has at least one dominant letter.
What? You got three green and red? I got two.
All right.
Oh, this one. Right here.
Another minute, and then we'll get going.
Yeah, there's three. And those, okay, yeah, okay. Yeah, you're here. Three, one, yeah. Did you get 1, 2, 3, 1? Yeah.
All right, what we got? 9 to 3 to 3 to 1?
Yeah.
That is correct. Okay. 9/16 show both dominant traits. 3/16 show one dominant and one recessive. 3/16 show the other dominant and the other recessive. 1/16 is pure recessive. Only has recessive alleles. Okay. 9 to 3 to 3 to 1. Now, there's a few things that we need to talk about. First of all, this is an independent assortment. What does that mean? It means, and Mendel didn't know this at the time, but it means that these two traits are in different chromosomes. So they don't have anything to do with each other as they go through meiosis. There's no crossing over. There's no mixing of any sort. They're completely have nothing to do with each other.
Yeah.
Whether or not you get yellow for seed color and whether or not you get wrinkled or round as a seed color, completely independent of each other. Okay, so that's an important point. But if it is independent assortment, okay, then your ratio, if you start off with two traits and you are heterozygous for both of those traits, you always get 9 to 3 to 3 to 1. Always. Now, I tell you this because you're probably going to get on the AP exam, and maybe on mine as well, a question that's going to ask you how many are going to be green and round. And if you have to sit there, it took a while, right, to do the Punnett square? It took some time. When you take the AP exam, it's time. And it's going to take some time to do a Punnett square. They assume that you know the shortcut, which is if these guys are both heterozygous, then you can use that shortcut of 9 to 3 to 3 to 1. Okay? Both heterozygous. And that is actually shown on that page. 9 to 3 to 3 to 1. Now, I don't want to touch on this too much today because I think it's enough just talking about the dihybrids right now. But occasionally, you have this happen where it's dependent assortment. And that's where they go as a package. They go as a package. So if you see one particular trait, you also see the other one as well. And that's because they're no longer on separate chromosomes. They're on the same chromosome. Okay? Let me give you a concrete example. Blonde hair. Usually also what? Blue eyes. Okay? Not always, but often, because that is dependent assortment. Those two genes are both found on the same chromosome. So if crossing over happens and somebody gets blonde hair, probably also the gene for blue eyes will also get passed over. So they always end up on the same chromosome. That doesn't happen if it's independent assortment. So you can start looking at that because what they like to give you is data, and they'll give you a whole bunch of data, and they'll say, does this show independent assortment or dependent assortment? And depending on what values you get, if you get 9 to 3 to 3 to 1, then nope, they're on different chromosomes. But if you get something different, if you get the offspring looking like one of the parents, but very few of the kind of hybrid ones that are in between that don't look like either parent, then that's dependent assortment, and you know that they're on the same chromosome. Right? Just think about that.
How do you do like dependent? Like how would you do that on a pi square? Or would you just not do that?
Well, you would have to do it a little differently, and that's what this is showing here. So you only get what the parents were. So you either get big R, big Y, big R, or small y, small r for both of the parents. And so you get completely different values. Okay. Interesting.
Oh, so the way you separate it.
Yeah, the way you separate it is going to be different. Yeah.
Okay. That's very weird.
All right, so let's see what we got here. We got anything else? Okay, Mendel's laws are governed by probability. We call this Mendelian genetics. Not everything exhibits Mendelian genetics, but it's the simplest and usually the go-to when you're teaching genetics. It's the first thing that introductory bio students get taught is Mendelian genetics. When tossing a coin, the outcome of one toss has no impact on the other. Oh, that isn't what I wanted. Okay. I can flip a coin, and that coin flip has nothing to do with another coin flip. Okay. That's the way it is with alleles, as long as they're on different chromosomes. Whether or not you get one allele has nothing to do with whether or not you get a different allele for another trait, because they're on different chromosomes. They're independent events. Some examples. What's the probability of a couple having a girl? Flip your coin. Oh, boy. Ah, dang it. Just, how many times do I have to hit that? Half. What's the probability of having a boy? Half. Okay. What's the probability of having two boys and a girl? Each event is completely independent of each other. So, probability of having a boy? 1/2. Probability of having a boy? 1/2. Probability of having a girl? So it's 1/8. 1/8. Oy. Oy vey. Okay.
He did not understand that shit.
Okay, let me do a little bit more and then we'll finish. Mendelian genetics is really simple, and in some ways Mendel got really lucky because the plants that he was using, the pea plants he was using, exhibited a very simple, easy, straightforward dominance and recessive. There was only 2 alleles for it, and you either were dominant or you were recessive, and that was it. But there's a lot of characteristics, traits that are not inherited by just one gene with two alleles. And so there's a lot beyond Mendelian genetics that we can talk about, and we will talk about. And let's look at, okay, degree of dominance. This is one thing that we can look at. Complete dominance means that you've got a dominant heterozygote completely overshadows the recessive. And the heterozygote looks identical to the homozygous dominant because there's no difference. It's complete dominance. There's also something called incomplete dominance. And that's where, when you have a heterozygous condition, it's something that's kind of in between. It's not really dominant. It's not really recessive. It's something in between. And then you also have an example called codominance. And codominance is where, so incomplete dominance is kind of where they blend together, where you see something like red and white flowers, if they're heterozygous, look what?
Pink.
Pink, yeah. Codominance is a little different. Codominance is where those alleles, there's not really a dominant, there's not really a recessive. When you put those two together, those two alleles together, they both show themselves equally. And the example of that would be blood type. This example is showing you incomplete dominance. Here I have red flowers and I have white flowers. And the way this has to set up is they'll tell you these are snapdragons, kind of plant, kind of flower, and they exhibit incomplete dominance. So once you usually assume complete dominance, but if they say incomplete dominance, then you're going to use a different set of rules. Okay. So in that case, you don't have a dominant and a recessive allele, and so we say they're using a C for color. I'm just going to use big R, big R. For red, it's a pure breeding red flower. It always produces red flowers. And then we also have a pure white. But instead of using a small r, we use a W because it's not a recessive trait. When you breed these guys, you get that. You get a big R and you get a big W. And the two colors blend together and you see pink. Okay, that's incomplete dominance. So then I could actually breed a couple of these guys. I do a Punnett square. I have a big R, big R, big R, W, or W, and W, W. So I'm going to have one red. Let's change our colors. I have two pink, and I have one white. Okay. So rather than heterozygotes showing the dominant trait, there is no dominant trait. They're showing a blend. They're showing a color that's halfway in between, which is pink. Okay. Classic example of incomplete dominance. Questions on that? Sorry, I should have just gone away. All right. Okay. Now, we will talk about codominance next time. I'm going to leave off on this slide, which is on the frequency of dominant alleles. A dominant allele means if it's present, it shows itself. It does not mean it's more common in the population. Okay, that's a very common misconception that they like to test you on, is the idea that, well, if it's dominant, everybody's got it. Okay, and there's lots of things that are dominant that most people don't have. For example, polydactyly is having an extra digit, extra finger, and extra toe, and it is a dominant trait. Okay, so if you have the allele, you're going to have an extra finger and extra toe. Anybody in here willing to tell if they have an extra digit? No? Yeah, probably not. I mean, even if you did, it'd be one person. It's saying here one baby out of 400. So it's pretty small. So dominant doesn't mean more common in the population. It just means that if it's in the mix with another allele that is recessive, it will show itself. Okay, it's going to dominate over that other allele. That is all. There's also things like Huntington's disease. Huntington's disease is also a dominant inheritance, so you only need that one dominant copy to get it. Very few people, fortunately, have Huntington's disease. All right, questions? Tomorrow, and by tomorrow I mean Monday, we will talk about blood types.